Welcome to the Deep Dive. Whether you're prepping for a high-level neurobiology meeting, or maybe you're a mother trying to understand your own cognitive changes, or just someone who's insanely curious about the outer limits of human neuroplasticity, we've built this specifically for you.
Yeah, we are tackling a topic today that touches the lives of, well, millions. It's something so universally recognized that it's embedded itself deeply into our everyday cultural lexicon.
I am talking about mommy brain.
Right. Or baby brain, as it's sometimes called.
Exactly. And if you have ever been pregnant or if you've spent any significant amount of time around someone who has recently had a child, you are intimately familiar with the subjective feeling of this phenomenon. You know exactly what it looks like.
It's that pervasive, intensely frustrating cognitive fog.
Yes, it's the sudden, bizarre forgetfulness where you, I don't know, find your car keys inside the refrigerator.
Right.
It's the distractibility, the classic feeling of walking confidently into a room and then standing there, completely black, wondering what on earth you came in there to do.
Yeah. It's a universally reported experience, and frankly, for decades, it was completely brushed off by the broader medical community.
Oh, totally dismissed.
Right. If a woman complained about this cognitive fog, the standard clinical response was to dismiss it as a mere byproduct of sleep deprivation.
Or physical exhaustion.
Exactly. The stress of a changing body, or just the sheer overwhelming nature of a major life transition. It was treated as a lifestyle symptom, not a biological reality. But, and this is the crux of it, over the last decade, the science of neuroimaging has finally caught up to the subjective experience.
Which brings us to our core mission statement for today. Our goal for this deep dive is to validate your curiosity about what is actually happening inside the maternal brain. But we are going strictly by the data.
Only the data.
Right. We are stripping away the usual anecdotes, the folklore, and the casual dismissals from well-meaning relatives. We're looking exclusively at the hard, objective neuroscience of cognitive performance. We want to know what is happening to the structural hardware and the functional software of the brain.
We are focusing entirely on executive function, cognitive control, working memory, and the physical, structural, and network changes that underlie them.
And to do this justice, we have assembled a massive stack of sources.
A mountain of high-quality neuroimaging data, really. We'll be looking at functional MRI studies that track the brain in real-time, structural MRI studies that measure the physical volume of brain tissue.
And resting state functional connectivity scans.
Yes. We are looking at systematic reviews and meta-analyses spanning over a decade of intense research. You will be hearing data from major peer-reviewed publications, Frontiers in Psychology, Neuroscience, and Biobehavioral Reviews, BMC Pregnancy and Childbirth,
And several landmark papers from the Nature portfolio.
It is a robust, deeply empirical stack of evidence.
But before we dive into that mountain of data, we need to lay down a very strict ground rule for today's conversation. We want to be explicitly clear with you right up front. We will not be talking about the emotional centers of the brain today.
No, we are completely skipping social cognition.
Right. You will not hear us mention the amygdala. We aren't diving into empathy networks. And we will not be discussing emotional processing or bonding.
It's a vital boundary to set. I mean, the maternal brain undergoes massive, highly documented changes in those emotional and social areas, and that evolutionary bonding process is fascinating. But today is a pure cognitive performance and executive function deep dive.
We are isolating the variables.
Exactly. We want to look strictly at the brain's computing hardware as it relates to memory, attention, organization, and executive control.
Okay, so the behavioral reality is where we need to start. We throw around these colloquial terms, pregnancy brain and baby brain, but I want to know what this actually looks like when we isolate the cognitive variables in a clinical setting.
Right. Getting past the subjective.
Yeah. We're talking about genuine forgetfulness, a tangible difficulty in concentrating for extended periods, and trouble holding new pieces of information in the mind while trying to execute a task. Is this just exhausted people taking tests poorly, or is there a baseline drop in pure cognitive ability?
Well, we no longer have to guess or rely on self-reported questionnaires to prove it exists. We can dive straight into the behavioral data, and the numbers clearly validate the experience. We can anchor this entire behavioral discussion on a phenomenal 2018 meta-analysis by Davies and colleagues. The paper is titled Cognitive Impairment During Pregnancy, a Meta-Analysis, and it was published in the Medical Journal of Australia.
Now, whenever we look at medical research, a meta-analysis is the gold standard, right? Because they aren't just looking at one group of 20 women in a single hospital.
Correct. A meta-analysis aggregates the raw data from multiple independent studies, filtering out the weak methodologies, to find the undeniable, overarching statistical truths across thousands of participants. The Davies team looked at a wide array of cognitive domains, comparing pregnant women directly to tightly matched non-pregnant control groups.
And they control for things like age and education, right?
Yes, they controlled for age, education level, and baseline intelligence.
So what did the Davies team actually find when they stripped away all those confounding variables?
The data is definitive and statistically significant. They found that general cognitive functioning, memory, and executive functioning are distinctly lower in pregnant women when compared directly to the non-pregnant controls.
Wow.
This isn't just a slight, unnoticeable dip. It is a measurable impairment across multiple complex cognitive domains. And what makes this paper so critical is that they didn't just treat pregnancy as a single, monolithic 9-month block.
Right, because things change so drastically month to month.
Exactly. They broke down the gestational timeline to see exactly when this cognitive decline begins to manifest.
I would assume it happens at the very end. When the physical burden of the pregnancy is at its absolute highest and sleep is severely compromised.
That is the logical assumption, but the data tells a different story. The meta-analysis revealed that memory performance, specifically, shows a marked and significant decline transitioning from the first trimester into the second trimester.
Really? That early?
Yes. And, more importantly, it doesn't bounce back. It establishes a new, lowered baseline and remains there straight through the third trimester. So, long before the extreme physical exhaustion of the late third trimester sets in, the objective cognitive impairment is already fully established and clinically measurable.
That is fascinating. So the behavioral data proves the fog is real, and it starts early. But I want to get much more granular here. Memory is a massive umbrella term. I want to dissect exactly which cognitive functions are taking the hit and which ones remain intact.
To get that level of granularity, we turn to a highly recent, comprehensive 2024 paper by Eunice et al. It's titled Exploring the Influence of Pregnancy on Cognitive Function in Women, a Systematic Review, published in BMC Pregnancy and Childbirth.
Okay, 2024, so this is cutting edge.
It is a goldmine for cognitive neuroscientists because it categorizes the specific types of memory and executive tasks that are affected. For instance, they look heavily at verbal recall.
So if I'm sitting in a clinic taking a verbal recall test, what am I actually being asked to do?
The UNI's 2024 review highlights several key studies, notably the foundational behavioral work by researchers like Glynn and DeGroot. These studies utilized rigorous objective testing, such as word learning tasks and paired associate learning tasks.
Paired associate.
Yeah. In a paired associate task, the clinician asks you to memorize pairs of entirely unrelated words. For example, apple and chair, or river and bottle.
Okay.
Later in the session, they will prompt you with the word apple, and you have to consciously recall the linked word chair. The data consistently shows that pregnant women score significantly lower on these specific verbal recall tasks compared to non-pregnant controls.
That makes a lot of sense when you think about the real-world complaints. It's not just forgetting a random thought. It is a measurable drop in the active ability to encode and retrieve new, specific verbal information.
Exactly.
But I hear the term working memory thrown around a lot in these discussions. How is that different from me remembering my childhood phone number or the plot of a movie I saw last week?
It's a crucial distinction in neuroscience. Think of your brain's memory systems like the architecture of a computer. Your long-term memory is the hard drive. It's where you store things permanently, your childhood memories, how to ride a bike, your home address.
We're the permanent archive.
Working memory, on the other hand, is like the computer's RAM, the random access memory. It's the active, temporary workspace of the mind. It is the mental scratch pad you use to hold information just long enough to manipulate it, use it, and then discard it.
So it's the difference between knowing the multiplication tables by heart, which is long-term, versus trying to hold a friend's phone number in my head while actively calculating a 20% tip on a dinner bill.
Exactly. Working memory is highly dynamic. It requires active manipulation and heavy oversight from the brain's executive centers. And what the Eunice 2024 review found is that working memory shows variable declines during pregnancy, but these declines become glaringly pronounced in tasks that require a very strong executive component.
Meaning what, exactly?
If a cognitive task requires you to not just hold a piece of information, but actively reorganize it, sort it, or filter out heavy environmental distractions while holding it, that is where the pregnant brain shows the most severe strain.
Which brings us right to the concept of executive functioning. If working memory is the active workspace, executive functions are the CEO of the brain. They are housed primarily in the frontal cortex. They regulate our attention, our planning, our impulse control, and our cognitive flexibility, which is our ability to switch seamlessly between different concepts or rules.
And this is where the UNIS 2024 review is particularly illuminating regarding the daily struggles of mommy brain. They found significant negative impacts on a metric called the speed of comprehension.
Meaning the raw processing power required to take in new, complex information, synthesize it, and understand what it means is measurably slowed down.
Yes. The accuracy might still be there, but the processing time is elongated.
Yeah.
Furthermore, and perhaps most relevant to the daily lived experience, the UNIS review found a distinct, highly measurable negative effect on prospective memory.
Perspective memory. I love this concept. This is essentially the cognitive act of remembering to remember, right?
That is the perfect way to describe it. Perspective memory is the ability to formulate an intention and then remember to execute that intended action at a specific point in the future.
Like taking a pill.
Exactly.
Yeah.
It's remembering that you need to take a specific prenatal vitamin at exactly 4 p.m., or remembering that you need to pick up a specific brand of milk on the way home from work, or remembering to attach a document before you hit send on an email.
That sounds simple, but cognitively it must be exhausting. You have to maintain a background awareness of a future goal while actively engaging in your current present moment tasks.
It requires an incredible amount of frontal cortex oversight to pull a future intention into the present moment at the exact right time without a physical trigger reminding you. And the clinical behavioral data clearly and consistently shows that perspective memory is significantly compromised during pregnancy.
Okay, so the behavioral data proves the fog is real. We have a clinical drop in verbal recall. Working memory is strained under high cognitive load, the speed of comprehension slows down, and perspective memory, remembering to remember, falters.
Yes.
But I am stuck on the why. Are we just looking at exhausted software here, or is the actual physical hardware of the brain changing?
That is the big question.
Right, because if my computer starts running slow, sometimes it's because I have too many tabs open, but sometimes it's because the physical hard drive is failing. What is physically happening to the brain's tissue to cause this measurable drop in performance?
To answer that, we have to look directly at the physical architecture of the brain. We have to examine structural neuroimaging. We're talking about mapping the actual physical volume and density of the brain's gray matter.
And how do they do that?
Researchers measure this using structural magnetic resonance imaging, or MRI, paired with an advanced computational technique called voxel-based morphometry.
Let's break down voxel-based morphometry so we all understand the sheer precision of what we are looking at. I know a pixel is a 2D square on a screen. Is a voxel just the 3D version of that?
Yes, precisely. A voxel is essentially a three-dimensional pixel. When neuroscientists run a structural MRI, the scanner maps the brain in millions of these tiny 3D voxels.
Okay.
Voxel-based morphometry, or VBM, is a statistical neuroimaging analysis technique that allows researchers to measure the exact volume, density, and concentration of brain tissue in every single cubic millimeter of the brain. It allows them to take a scan of a woman's brain before she gets pregnant, take another scan during or after pregnancy, and calculate exactly where the physical tissue has changed down to the millimeter.
That is incredible precision. So what happens when you put a pregnant woman's brain through a VBM analysis?
The findings generated by this technology over the last decade have completely upended our understanding of adult human neurobiology. We have to introduce the landmark findings discussed in two absolutely pivotal papers.
Let's hear them.
The first is a highly recent 2024 paper by Pritchett et al., titled Neuroanatomical Changes Observed Over the Course of a Human Pregnancy, published in Nature Neuroscience.
Huge journal.
The second is the foundational, groundbreaking 2016 paper by Hoeksema et al., titled Pregnancy Leads to Long-Lasting Changes in Human Brain Structure, which was also published in Nature Neuroscience.
I mean, Nature Neuroscience is the absolute pinnacle of scientific publishing. You don't get in there with small sample sizes or weak statistical correlations. So what did these two landmark studies physically observe in the hardware of the maternal brain?
The key detail from both of these studies is staggering in its consistency and its magnitude. Let's look at the methodology of the Hoeksema paper because it really set the standard for this entire field. They didn't just scan women once they were already pregnant, they recruited women who were planning to become pregnant and scanned their brains before conception.
Oh, that's smart.
Then they waited. Once the women had their babies, they brought them back and scanned them again. And crucially, they didn't just compare them to non-pregnant women. They compared them to fathers and to men who are not fathers to ensure that any brain changes weren't just the result of the lifestyle shift of having a crying newborn in the house.
That is a brilliant control group. You isolate the biological variable of gestation from the environmental variable of parenting.
Precisely. And what they found was that primiparous mothers, meaning women going through their first pregnancy, undergo a highly consistent symmetrical pattern of extensive gray matter volume reductions across the gestational period.
Reductions?
Yes. The structural changes were so distinct, so uniform, and so specific to the pregnant women that the researchers could use a computer algorithm to predict with 100% accuracy whether a woman had been pregnant or not, purely by looking at the raw VBM data of her brain scan.
Wait, hold on. You're telling me the brain is physically shrinking?
Yeah.
The gray matter volume is reducing?
Yes.
If my doctor told me that a major organ in my body was physically losing volume, I would panic. That sounds like atrophy. That sounds like the cognitive damage you see in early-stage dementia or Alzheimer's.
That is the immediate knee-jerk reaction for most people, and even for many clinicians who first read this data. When we hear gray matter volume reduction, we are culturally conditioned to equate it with brain damage, pathological loss, or decay.
Naturally.
But we have to look specifically at the geography of these reductions because it ties directly back to the behavioral data we just discussed. This extensive gray matter loss heavily impacts the bilateral, lateral, prefrontal, and temporal cortices.
The prefrontal cortex. That is the exact anatomical home of the executive functioning working memory, prospective memory, and cognitive control that the behavioral tests showed were impaired. The physical computing nodes responsible for those specific cognitive tasks are actually shrinking in volume.
Yes, the volume is physically reducing, but here is where we need to entirely reframe this concept of loss. To understand why this isn't dementia, we look at an incredible 2022 review by Polisky et al. It is beautifully titled, "Less Can Be More Fine-Tuning the Maternal Brain," published in Neuroscience and Biobehavioral Reviews.
Less can be more. I like that.
The authors provide an in-depth analysis of a vital biological mechanism known as experience-dependent plasticity and synaptic pruning.
So if it's not atrophy, what is synaptic pruning? How does losing brain tissue make a brain better?
Think of a wildly overgrown garden. It has leaves, vines, and branches sprawling everywhere. It looks incredibly lush and dense, but because it is so overgrown, the sunlight can't penetrate to the lower levels, the roots are tangled, and the limited nutrients from the soil are spread far too thin across thousands of unnecessary weak shoots. The garden is big, but it is highly inefficient. Now imagine a master gardener comes in and aggressively prunes that garden. They cut back the sprawling branches, remove the excess foliage, and strip away the weak vines. If you look at the total volume of plant matter, the garden technically has less. It has shrunk.
But the remaining plants are vastly stronger.
Exactly. The nutrients are hyper-focused into the core fruit-bearing branches. The pruning wasn't damage, it was a necessary, deliberate refinement.
So the pregnant brain is acting like that master gardener. It is actively shedding excess synapses and neural connections that it no longer needs to create a more streamlined, hyper-efficient neural architecture. But what triggers the gardener to start cutting?
The primary triggers are the massive, unprecedented hormonal cascades of gestation. During pregnancy, a woman's body produces an unparalleled surge of sex steroid hormones, particularly estrogen and progesterone. To give you a sense of scale, a woman produces more estrogen during a single pregnancy than she will throughout the entire rest of her life combined.
That is wild.
And these hormones are not confined to the reproductive organs. They readily cross the blood-brain barrier.
And once they cross into the brain, what do? Do they act as the shears?
They act as the master architects, signaling the local immune cells of the brain called microglia to begin the pruning process. The reduction in gray matter that Prisha and Hoxham observed is not brain damage. It is a highly deliberate, hormonally driven, evolutionary fine-tuning of the female brain.
It's adapting.
It represents a transition from a generalized, all-purpose neural state into a highly specialized state optimized for the immense cognitive demands of keeping a highly vulnerable human infant alive. In neurobiology, we compare this dynamic developmental phase, often termed matrescence, to the only other time in a human lifespan when we see such massive, rapid, hormonally driven neural remodeling.
Adolescence.
Exactly. During puberty, the adolescent brain undergoes an intense wave of synaptic pruning, aggressively shedding gray matter volume to transition from a child's brain into a biologically mature adult's brain.
It's chaotic, it affects behavior, but it's totally necessary for maturation.
Right. Pregnancy triggers a second, distinct, and equally massive wave of synaptic pruning, transitioning the adult brain into a specialized maternal brain. It is an evolutionary upgrade, not a deficit.
That is such a profound paradigm shift. The gray matter, the actual computing nodes, are being aggressively pruned for long-term efficiency.
Yeah.
But if the physical computing nodes are shrinking, what is happening to the wiring that connects them?
That brings us to another brilliant and critical insight from the recent Pritchett 2024 paper. Everything we have discussed so far has been about gray matter, which primarily contains the cell bodies of the neurons, the processing centers, but the brain also has a massive network of white matter.
And white matter is the cabling, right?
Yes, white matter is made up of the myelinated axons, the physical insulated cables that connect different brain regions across long distances and allow them to communicate.
And what did the Pritchett team find when they analyzed the white matter of pregnant women?
They found that running completely parallel to the gray matter volume reductions, there are distinct, highly significant increases in white matter microstructural integrity.
I know they measure this using a highly specific physical metric called anisotropy. What does an increase in anisotropy actually mean for the physical cables in the brain?
We measure it using fractional anisotropy, which tracks how water molecules diffuse along the neural pathways. Without getting too deep into the quantum physics of the MRI scanner, an increased fractional anisotropy essentially means that the white matter tracks are becoming more highly organized, structurally sound, thickly myelinated, and strongly directional.
So it's an upgrade.
It is the neurological equivalent of taking a winding, pothole-filled, two-lane country road and systematically upgrading it into a high-speed, multi-lane fiber optic superhighway.
So if we synthesize these hardware changes, the massive hormonal surge of pregnancy is signaling the brain to aggressively prune away excess gray matter in the prefrontal cortex to make the computing nodes highly specialized and efficient, while simultaneously heavily upgrading the white matter cables to ensure lightning-fast, highly reliable communication between those specialized nodes.
Precisely.
It is a total architectural renovation of the executive system.
It is a phenomenal feat of biological engineering. But this architectural renovation poses a very difficult, very tangible physiological challenge for the woman experiencing it. While the brain is right in the middle of this massive construction project, paring down gray matter and laying down new white matter highways, the woman does not get to pause her life.
Right, she still has to function.
She still has to go to her job, she still has to solve complex analytical problems, she still has to navigate her daily social life, and she still has to rely heavily on her working memory.
Which brings us to a really crucial pivot in our deep dive. We are moving from structure, the physical shape, density, and volume of the brain, to function. I want to know how this newly pruned, under-construction brain actually operates in real time when it is suddenly challenged with a highly demanding cognitive task. Does it crash?
To investigate the real-time functioning of the brain, structural MRI is no longer sufficient. Structural MRI just gives us a beautiful, static 3D photograph of the brain's anatomy. To see the brain in action, we have to transition to functional magnetic resonance imaging, or fMRI. Functional MRI measures the blood oxygen level dependent signal, or the BOLD signal.
Explain the BOLD signal, because when people say a brain region lights up on a scan, they usually don't know what that actually means physiologically.
Right. When a specific network of neurons in the brain fires, it requires an immediate influx of energy to sustain that electrical activity. The body responds by rapidly rushing oxygen-rich and glucose-rich blood to that exact millimeter of tissue. Oxygenated blood has different magnetic properties than deoxygenated blood.
So the magnet detects the blood flow.
The fMRI scanner is essentially a giant magnet that detects those microscopic, localized changes in blood flow and oxygen consumption at a millisecond level. So when we say an area lights up, we mean that specific network of neurons is working so hard that it is demanding a massive surge of metabolic fuel.
And to see how the maternal brain handles a heady cognitive load while under construction, we're looking at an incredibly precise 2020 paper by Bok-Ak et al., titled altered neural substrates within cognitive networks of postpartum women during working memory process and resting state. How did they stress test these women?
The methodology is brilliant. They took a group of postpartum women and a tightly matched control group of non-postpartum women, put them into the fMRI scanner, and asked them to perform a classic cognitive assessment known as the N-back task.
Oh, I've heard the N-back task described as the absolute gold standard for testing working memory in cognitive neuroscience. I've taken one online, and it is infuriatingly difficult.
It is very tough.
For anyone who hasn't experienced it, how does it actually stress test the prefrontal cortex?
It's essentially the ultimate stress test for your brain's RAM. Imagine you are lying entirely still inside a loud MRI scanner, staring up at a small screen. A sequence of letters flashes on the screen, one by one, with a short gap between them. A, C, B, A, C. In a baseline one-back task, you simply have to press a button if the letter you are looking at right now is the exact same as the letter that appeared immediately before it.
That's relatively easy.
But in a two-back task, the cognitive load doubles. You have to press the button if the current letter matches the one that appeared 2 steps ago. In a 3-back task, it's 3 steps ago.
It forces you to constantly hold a rolling, continuously updating sequence of items in your active working memory, update the sequence every single second, discard the old information, and make an immediate executive decision based on that temporarily held data.
It is exhausting just thinking about it. It places a severe, sustained metabolic demand on the prefrontal cortex. Now, behaviorally, the results of the Back to 2020 study were fascinating, and they push back against the narrative that the maternal brain is broken. When looking purely at the scores, the actual behavioral output of the N-back task, the postpartum women performed just as well as the control group. Their accuracy was statistically equivalent. I got the answers right.
So the cognitive fog isn't making them fail the test.
No. However, the researchers noted a critical detail. The postpartum women took slightly longer to respond. Their reaction times were prolonged. They arrived at the correct answer, but the raw processing speed to get there was measurably slower.
Which perfectly aligns with the behavioral data from the UNIS review about a slower speed of comprehension. But the behavioral score is only half the story here. While these women were slowly but accurately passing the test, what did the fMRI scanner show happening to the blood flow inside their brains?
This is where we see the hidden physical cost of the hardware changes. During the heavy cognitive load of the working memory task, the postpartum women showed massive, statistically significant hyperactivation in the regions of the task-positive network, or the TPN.
What exactly is the task-positive network?
The TPN is a distributed web of brain regions, heavily featuring the lateral prefrontal cortex and parietal regions. It's the network that fires up whenever your brain's CEO takes charge. It activates when you are engaged in an attention-demanding, externally focused cognitive task, like doing complex math, reading a dense legal document, or performing an N-back test.
Okay.
In the postpartum women, the areas of the TPN were lighting up with far more intensity and consuming vastly more oxygen and glucose than the exact same areas in the non-postpartum control group.
Here's where it gets really interesting. I want to make sure I am conceptualizing this correctly. The postpartum women got the exact same score on the test as the control group. But to achieve that identical score, their prefrontal cortex had to demand significantly more blood, more oxygen, and more energy. They had to work much, much harder to get the same grade.
Exactly. This introduces one of the most vital neuroscientific concepts in this entire deep dive neural compensation. Because the prefrontal executive networks have been physically reorganized, pruned, and reduced in volume by the gestational hormones, those remaining neural circuits have to work significantly harder. They have to hyperactivate to achieve the same behavioral output on a demanding cognitive test.
Let's go back to our computer analogy. Imagine you have a computer processor that is right in the middle of a massive background system update.
Yeah.
Deleting old files, rewriting core directories, and optimizing the registry. That is the biological equivalent of the synaptic pruning and architectural reorganization of pregnancy. Now, if you suddenly force that computer to simultaneously render a complex, high-definition 3D video, which is the N-back task, the processor is going to struggle immensely. To manage that heavy, active workload without crashing the whole system, the computer has to run its cooling fan on absolute maximum speed, it draws maximum electrical power, and it pushes its remaining available hardware to the absolute thermal limit.
That is a phenomenal analogy for neural compensation. The maternal brain is successfully compensating for its altered, pruned hardware state, but doing so requires a massive sustained expenditure of metabolic energy.
Wow.
That extra energy requirement, that physical hyperactivation of the task-positive network, is almost certainly the biological root of the subjective sensation of brain fog, or the intense mental fatigue that so many mothers report. They can still do the complex math, they can still manage the intense project at work, and they can still pass the cognitive tests. But it feels deeply exhausting because their task-positive network is quite literally running in overdrive, burning more physical fuel to compensate for the ongoing structural remodeling.
It perfectly explains the lived experience. It explains why a daily task that used to feel effortless, like managing a busy schedule or organizing a grocery list, suddenly feels like you are pushing a massive boulder up a steep hill. Your brain isn't failing, it's actively burning more physiological fuel to get the exact same job done.
Perfectly said.
Okay, so we have a beautifully detailed picture of what happens when the maternal brain is under heavy cognitive load. It hyperactivates the task-positive network to survive the stress test. But humans are not constantly doing in-back tests. What happens when the brain is just chilling? What happens when a pregnant or postpartum woman isn't focusing on a complex task, but is just staring out the window, folding laundry on autopilot, or lying in bed trying to fall asleep?
To understand the resting brain, we have to look at the functional counterpart of the task-positive network. When the brain is not actively focused on a demanding external task, it shifts its energy into a completely different web of structures known as the default mode network, or the DMN.
The DMN.
Yes, the default mode network is the brain's baseline resting state. It becomes highly active when we are daydreaming, mind-wandering, recalling past autobiographical memories, thinking about the future, or engaging in internal self-referential thought.
It is the brain's internal idle mode. But given everything we've discussed about the hardware changes, I am guessing the idle node in the maternal brain is fundamentally altered.
It is deeply altered. To explore the resting maternal brain, we look at a highly precise 2018 paper by Zhang et al. It is titled Disrupted Spontaneous Neural Activity Related to Cognitive Impairment in Postpartum Women, published in Frontiers in Psychology.
Okay, so how do they test the resting state?
This research team didn't use an NBAC task to stress the brain. Instead, they used resting state fMRI, or RS-fMRI, to simply observe the spontaneous baseline metabolic activity of the brain, while the women were instructed to lie perfectly still, close their eyes, and think of nothing in particular.
So they're mapping the baseline hum of the brain. How do you actually measure that mathematically?
The Zhang 2018 study used two highly advanced neuroimaging metrics to quantify this resting activity. ALFF, which stands for Amplitude of Low-Frequency Fluctuation, and ReHo, which stands for Regional Homogeneity.
I love these metrics, but they can be incredibly dense to parse.
Yeah.
Let's translate ALFF and ReHo into a quick, relatable analogy.
Let's do it.
Imagine the resting baseline brain is a giant, complex choir humming a specific tune. ALFF, the amplitude of low-frequency fluctuation, is essentially measuring the volume of the raw intensity of the local brain activity. It asks, how loud is a specific section of the choir singing right now?
Right.
ReHo, on the other hand, regional homogeneity, is measuring the localized synchronization. It asks, how well are the neighboring neurons, the individual singers, standing next to each other in that section of the choir firing in sync? Are they harmonizing perfectly, or is it a chaotic, disjointed noise?
That is an exceptionally clear way to explain complex Bold-D signal analytics. So the Zhang team mapped the resting volume, ALFF, and the resting synchronization, ReHo, of the postpartum brain, and what they found was highly significant.
What was it?
The study revealed distinctly decreased spontaneous neural activity in highly specific nodes of the default mode network. Primarily, they saw significant drops in activity within the posterior cingulate cortex, or the PCC, which acts as a massive central hub of the DMN. They also observed heavily decreased baseline activity in the prefrontal cortex, specifically targeting the superior frontal gyrus and the middle frontal gyrus.
So to use our analogy, the volume of the choir is turned down in the prefrontal cortex, and the singers in the posterior cingulate cortex are out of sync. But does a disruption in the resting state actually impact how a person functions? Did the Zhang team correlate these resting brain scans with actual measurable behavioral tests?
They did, and the correlations were striking. They found that the specific decreases in the PCC and prefrontal resting activity strongly and negatively correlated with poor performance on two specific, highly rigorous cognitive assessments: the complex figure test, specifically the delayed recall portion, and the clock drawing test.
The clock drawing test sounds simple, but I know it's used extensively in neurology to detect subtle cognitive impairments. And the complex figure test is brutally difficult. Can you detail the intense cognitive demands of these specific tests?
Certainly. The complex figure test requires a person to look at a highly intricate, abstract geometric line drawing, one with dozens of intersecting shapes, lines, and details. First, they must copy it while looking at it. Then the drawing is taken away, and after a significant time delay, they must reproduce that highly complex image entirely from memory.
Well, looking.
Yes. The clock drawing test asks the patient to draw the face of an analog clock from scratch, put in all the numbers in the correct positions, and set the hands to a specific, potentially confusing time like 10 past 11.
These are not tests of how tired you feel. These are rigorous objective measures of highly complex visual spatial memory, visual constructional skills, and high-level executive function, planning, and organization. You have to hold rules in your head, access spatial memory, and execute a motor plan simultaneously.
Exactly. And the Zhang 2018 study provides the direct, undeniable neurobiological link. The more disrupted the spontaneous resting activity was in a woman's PCC and prefrontal cortex, the worse she performed on those complex executive and visuospatial tasks. It definitively proves that the brain fog isn't just a byproduct of being distracted by a crying baby. It is directly, physically correlated with measurable baseline disruptions in the spontaneous neural activity of the brain's core resting networks.
Now, this entire discussion, so far from the pruning of the gray matter to the hyperactivation of the task-positive network to the disruption of the default mode network, has focused heavily on premiparous women. First-time mothers undergoing this monumental, hormonally driven biological shift for the very first time. But a natural, vital question arises, and I know many listeners are wondering this right now. What happens if you do it again? What happens when you have baby number 2? Does the brain undergo the same massive pruning and disruption all over again, or is the architecture already permanently set from the first round?
It's a critical question, and frankly, it is an area of neuroscience that is only just beginning to be explored with high-resolution resting state imaging. But we have emerging data. We can look to a very recent 2022 paper by Zhang et al. Titled Decreased Brain Functional Connectivity Associated with Cognitive Dysfunction in Women with Second Pregnancy, published in Frontiers in Aging Neuroscience.
Oh, yeah.
This study specifically looked at parous women, meaning women who were currently on their second pregnancy, and compared their neuroimaging directly to premiparous women on their first pregnancy and nulliparous controls who were never pregnant.
And when the Zhang team talks about functional connectivity, how is that different from the ALFF and REHO metrics we just talked about?
Where ALFF looks at the volume of localized activity, functional connectivity looks at the strength of the long-distance communication lines. It measures how well different, geographically separate regions of the brain communicate and synchronize with each other during that resting state. How efficiently is node A talking to node B across the brain?
So how did the communication lines fare in the women on their second pregnancy?
The key findings of the Zhang 2022 study were fascinating. They found that parous women on their second pregnancy showed distinctly disrupted functional connectivity entirely within the default mode network.
Within the resting network again?
Yes. Specifically, the researchers measured a significant decrease in the functional connectivity between the posterior cingulate cortex, the parahippocampal gyrus, and the prefrontal cortex. The core communication lines between these absolutely crucial memory and executive hubs were measurably weakened.
The parahippocampal gyrus is heavily involved in memory encoding and retrieval. So if the communication line between the memory center and the executive prefrontal center is disrupted during a second pregnancy, I have to assume that correlates to a drop in behavioral performance. Did they test them?
They absolutely did test them. The researchers administered the digit span test, which is a classic foundational clinical test of short-term and working memory. The clinician reads a sequence of numbers, and the subject has to repeat them back, first forward and then in reverse order, with the sequence getting progressively longer.
Very standard.
The study found that the specific decrease in functional connectivity between the PCC, the parahippocampal gyrus, and the prefrontal cortex was directly, significantly linked to impaired short-term memory on the digit span test, as well as broader executive dysfunction.
So if we synthesize all of this, whether it is the massive architectural shift of the first pregnancy or the ongoing functional disruptions of the second part, the clinical data paints a remarkably consistent picture. The intense, unprecedented hormonal cascades of gestation trigger massive architectural pruning in the prefrontal cortex.
Yes.
This loss of gray matter hardware forces the brain's task-positive network to severely hyperactivate, burning immense energy during complex cognitive loads just to maintain a baseline level of performance. Simultaneously, this structural renovation disrupts the baseline resting activity and the long-distance connectivity of the default mode network. The end result is the direct biological manifestation of mommy brain, the strained working memory, the slowed processing speed, and the undeniable executive fog.
It is a profound, systemic, brain-wide biological reorganization. But stopping the analysis here leaves us with a critical, almost existential physiological question about the lifespan of the female brain.
Right. If the brain permanently loses gray matter volume during pregnancy and the resting networks become fundamentally disrupted, we have to pose this crucial question: does the brain stay this way forever? Are these women permanently operating with a hyperstrained, pruned-down prefrontal cortex for the rest of their lives? To go back to our analogy, does the pruned garden ever regrow its leaves, or is the sheer volume of the brain permanently diminished?
To answer that, we have to transition away from pregnancy and look at the science of postpartum plasticity. And I am thrilled to say that the data here is incredibly dynamic, hopeful, and frankly, astonishing. We looked to a fascinating 2020 paper by Luder et al., evocatively titled "From Baby Brain to Mommy Brain, Widespread Gray Matter Gain After Giving Birth," published in the journal Cortex.
Cortex is great.
We also heavily rely on foundational longitudinal work from a landmark 2010 study by Kim et al., published in Behavioral Neuroscience.
Now, these postpartum studies took a slightly different methodological approach to the timeline, right?
Yeah.
Instead of just comparing a scan from a year before pregnancy to a scan a year after, they zoomed in specifically on the immediate postpartum window.
Yes, the timing of the scans is the genius of these studies. They utilized high-resolution structural MRI to look at the brain in incredibly tight specific timeframes. The researchers captured scans of the women's brains almost immediately after childbirth within the first 1 to 2 days postpartum, while they were often still in the hospital.
Wow, right after birth.
Then, they meticulously tracked those exact same women and brought them back for follow-up structural scans weeks or months later, typically mapping the brain at 4 to 6 weeks postpartum, or 3 to 4 months postpartum.
They are tracking the brain's physical volume week by week in the immediate aftermath of birth.
Yeah.
And what did the structural scans reveal?
The key findings are an absolute revelation in adult neurobiology. When the researchers compared the structural MRIs taken immediately after birth at day 1 or 2 to the scans taken months later, they did not see further reductions. They did not see the volume stabilizing at the newly pruned baseline.
They saw growth.
They saw widespread, highly significant increases in gray matter volume. The maternal brain was actively, rapidly rebuilding physical neural tissue.
It's regrowing the tissue. It is a remarkable display of adult neuroplasticity. We are taught that once you hit adulthood, your brain is essentially fixed, and you just slowly lose neurons as you age. But this proves the adult female brain can generate massive structural growth. Where exactly in the brain is this volumetric rebound happening?
We must be specific about the geography of this regrowth because it is not random. The researchers detailed that these significant rapid gray matter gains occur highly specifically in the prefrontal cortex and the parietal lobes.
Let me stop you right there. The prefrontal cortex and the parietal lobes. Those are the exact neural homes of cognitive control, working memory, and executive function. Those are the exact geographical areas that were heavily pruned and reduced during the gestational period by the hormonal surges.
Exactly. The very areas that were torn down are the ones being intensely rebuilt. This reveals that the transition to motherhood is not a single event. It is a biphasic, or two-part, neuroplastic journey of extreme proportions. If we synthesize the entire timeline across all these massive Nature, Cortex, and Frontiers papers, the story of the maternal cognitive brain is arguably the greatest masterclass in biological adaptation in the human lifespan.
Let's trace that entire timeline from start to finish to really cement how incredible this is. Phase 1 is gestation.
Yes. Phase 1 pregnancy. The body produces an unparalleled surge of estrogen and progesterone. These hormonal surges cross the blood-brain barrier and act as master signaling molecules. They trigger a massive, highly targeted wave of synaptic pruning. The local immune cells aggressively reduce the gray matter volume in the prefrontal cortex and temporal cortices. Again, this is not pathological damage. It is a highly evolved, fine-tuning mechanism designed to clear out old, inefficient neural pathways to prepare the brain for a massive paradigm shift.
But because that essential hardware has been aggressively pruned, the immediate side effect, the biological cost, is that the woman experiences a temporary, highly measurable strain on her working memory and her processing speed. When she tries to execute complex cognitive tasks, her brain is forced to rely on intense functional compensation. Her task-positive network has to hyperactivate, burning vastly more metabolic energy, making cognitive effort feel significantly more exhausting. Meanwhile, her resting state default mode network experiences transient disruptions in its baseline connectivity. The fog rolls in. That is the biological, undeniable reality of pregnancy brain.
Perfectly summarized. Then comes the physiological event of childbirth, and we violently enter phase 2, the postpartum rebound. Once the baby is delivered and the placenta is expelled, two monumental things happen to the mother simultaneously. First, those extreme gestational hormone levels, the estrogen progesterone that drove the pruning, plummet rapidly back toward their pre-pregnancy baselines. The pruning signals stop.
And the second thing.
Second, the mother is immediately thrust into one of the most cognitively demanding, high-stakes, hypervigilant learning environments a human organism can possibly experience, keeping a highly vulnerable newborn alive.
She is engaged in intense 24/7 life-or-death experiential learning while severely sleep-deprived.
Exactly. And the brain responds dynamically to that incredible, unyielding environmental demand. Driven by the stabilizing hormone baselines and the intense, continuous sensory stimuli of mothering, the brain undergoes a second massive wave of neuroplasticity. But this time, the plasticity is additive.
Building up.
The brain begins to rapidly rebuild, synthesize, and increase gray matter volume right back in the prefrontal cortex and parietal lobes. It is essentially growing brand new, highly optimized neural hardware to master a massive new repertoire of highly complex executive behaviors.
It's building specialized hardware for motherhood. It's growing new volume to manage complex, fragmented sleep schedules, to track intricate feeding times, to accurately predict the needs of a completely nonverbal infant, and to maintain a state of continuous background hypervigilance while still managing her own survival.
It is a breathtaking biological process when viewed in its totality. The maternal brain quite literally tears itself down to its studs during pregnancy to get rid of the old, generalized wiring, and then rapidly builds a brand new, hyper-efficient, specialized executive network in the postpartum period to manage the infinite cognitive complexities of motherhood.
Okay, let's take a deep breath. We have covered a monumental amount of hard neuroscience today, from voxel-based morphometry to functional network connectivity. So what does this all mean for you? If you take absolutely nothing else away from this deep dive, let it be this core truth: mommy brain is not a myth. It is not an excuse. It is not simply the result of sleep deprivation, and it is absolutely, categorically not brain damage or early cognitive decline.
Absolutely not.
It is the objective behavioral byproduct of one of the most radical, dynamic, and purposeful periods of neuroplasticity a human being can possibly experience. It is a literal, physical, hormonally driven rewiring of the prefrontal cortex and the working memory networks, designed entirely to optimize a woman's brain for a completely new phase of evolutionary survival.
It is a profound testament to the incredible resilience, plasticity, and adaptability of human biology. But before we conclude today, observing this entire neurobiological journey leaves us with a final, mind-expanding, somewhat provocative idea to mull over.
Let's hear it.
We have established, purely through the data, that the brain aggressively prunes itself during pregnancy and then rapidly rebuilds itself postpartum. We know this process temporarily strains working memory in order to create a highly specialized, hyper-efficient executive network.
Right. The overgrown garden is harshly pruned back, the soil is enriched, and that is specifically regrown to bear vastly more fruit.
Exactly. But let's extrapolate this massive neuroplastic event across a woman's entire lifespan. If this maternal brain is functionally equivalent to an intensely upgraded, streamlined operating system with newly laid white matter superhighways and highly optimized gray matter computing nodes, how might these long-lasting structural upgrades and enhanced functional connectivities play out decades later? Could this intense biological process ultimately act as a neuroprotective shield against cognitive decline as women age? Could the very intense, demanding neuroplastic teardown and rebuild that causes the temporary fog of baby brain in a woman's 20s or 30s be the exact same physiological process that builds an invincible, highly resilient cognitive reserve that protects her against dementia or neurodegeneration decades later in her 70s and 80s?
That is a staggering thought to end on. The idea that the frustrating, exhausting cognitive fog of today is actually the biological cost of building the impenetrable cognitive armor of tomorrow. The neurological literature is just beginning to hint at this long-term protective effect, and it completely, fundamentally reframes how we should view the entire maternal cognitive experience. You are not losing your mind. You are systematically upgrading it for the long haul. Thank you so much for joining us on this deep dive into the maternal brain. We hope this armed you with the data you need to understand the incredible shifts happening beneath the surface. Keep exploring the research, keep questioning the old narratives, and never stop marveling at the incredible, ever-changing architecture of the human brain. Catch you next time.